Hi, Namaskar. This is Dr. M. Arumugham, Assistant Professor and Head, PG and Research Department of Botany, JJ College of Arts and Science, Autonomous, Pudukottai, Tamil Nadu, India. In this video, we are going to see about the yes, the steps involved in the micropropagation technique. The fourth step is we have to clean and prepare the glasswares and prepare Newton medium. We have to select this explant and we have to sterilize it, and we have to prepare the inoculation for the aseptic medium, and preparation of shoot multiplication medium and transfer of shoots in subculture and root and hardening of plants and field trials. These are the protocol what we have elaborately discussed in the last video. Now I want to emphasize the culture methods. The first method is direct organogenesis and indirect organogenesis. The direct or okay. Then after the 6 to 8 weeks, by addition of BAP or any cytokinin along with Newton medium, we can have the elongated shoot, and it should be transferred to in vitro medium along with some oxygen supplemented Newton medium. If you wait 4 to 6 weeks, you can have some rooted plantlets. If you have a indirect organogenesis, means any explant, it may be cotyledonal leaves or it may be a shoot or a node or internode or hypocotyl, whatever it is, you have to undergo a de-differentiation. This is called callus induction. The callus induction may be done with the nutrient medium along with um, 2,4-D. If you wait 6 to 8 weeks, you can have the proliferated good callus in it. So the callus can be used for the various purposes. You can for the cell culture, you can go for the cell differentiation, or you can go for organogenesis, or you can go for the somatic embryogenesis. Whatever you want, you can go to that for the callus. From the indexed callus, if you want to organize a shoot system, you have to add the medium along with kinetin or any other proportion of the cytokinin. If you wait for 6 to 8 weeks, 4 to 6 weeks, you can have very good elongated shoots. That shoots will be transferred to the in vitro medium, what I already told you. And another one is direct somatic embryogenesis. The induction of embryos by the somatic cells like from the callus. So for induction of the somatic embryogenesis, we can go for shoot system or any cotyledon or leaf, nodes or any hypocotyl. So for induction of the somatic embryo, you have to add by BAP as well as kinetin with the different proportions along with the Newton medium. If you wait at least 8 weeks, you can have the somatic embryo induction or initiation. After it will be transferred the somatic embryo regeneration medium, which is supplemented with kinetin with MS medium of Newton medium. You can have the duration from 4 to 6 weeks. You can get the good somatic embryos. The somatic embryos, you can get the regeneration of shootlets, shoots, plantlets. So the acclimatization process may be at least 4 weeks for serving the good ratio. First, what is the explant? What I already told you, the explant is nothing but the excised portion of a parent or donor plant. It may be a shoot like a leaf, meristem, or shoot tip, or node, internode, bud, whatever it is. So the shoots develop from a very small fragment of a DNA. We'd have if an example of shoot apical meristem, it may be the ranging of 0.1 to 1.0 millimeter in length. You can go for the disease free plant. As like the meristem tip culture, there is a very good protocol. You have to select the meristem tip or meristem tip culture or meristem region. You have to go for the culture which is liquid medium. In the liquid medium we have to put this boat like Wattman #1 filter paper boat like structure. On the boat like structure we have to place that shoot tip. From the shoot tip we can have the new shootlets. So as indicated the image or the illustration, we can come to know that. Axillary bud. The node or axillary bud consists of the piece of stems with axillary bud with or without a portion of shoot. When only one axillary bud is cultured, is designated as axillary bud culture. If you are going to culture along with the shoot system, the like node or internode with the axillary bud, that culture as node culture or internode culture or whatever it is. What is cell suspension culture as callus culture? Once the plant parts such as a leaf disc, intercalary meristems, or any piece of stem, immature embryos, anthers, pollen grains, or any ovule have cultured to initiate the callus. So if you want to culture the callus, you can take whatever the explant you want. As rightly pointed out this illustration or the picture, the adult plant embryo culture, organ culture, callus culture, cell culture is having the different pathway or a protocol for production of new plantlets. The callus culture is concerned the initiation as a continued proliferation of the undifferentiated parenchyma cells. Ultimately, the callus meaning is undifferentiated, continuously dividing mass of parenchymatous tissues is called as callus. So undifferentiated, continuously dividing the parenchymatous cells may differentiate based upon the nutritional value or nutrients of the MS medium. As per the suspension is culture is concerned, the cell or group of cells dispersed and growing in a aerated liquid culture medium is placed in a liquid medium and shaken vigorously, and balanced nutrients and hormone ratio also, The cytokine will be induced advantageous bursts. For an example, in kiwi fruit, in a suspension culture, we can have the subculture for about 1 week. The pollen culture is taken from the pollen grains which germinated in vitro. Such cultures may eventually from the monohaploid, that is monoploid culture, from which shoots embryoid developed into the monoploid plants. What about the ovule culture? You know pretty well, the ovule is the female reproductive organ. The female reproductive gametophytic cells are also source for the haploid production, as like the pollen grain production or pollen grain culture. The ovule culture also a source of haploid production. For haploid production, the female gametophyte is necessary to know that one is events related to the induction of the haploid plant in these tissues. The image is rightly pointed out. The ovule, which is marked out, which is going to culture for the haploid production. In the case of shoot tip culture, the culture of the terminal point of the shoots of the plant into a culture environment or culture medium or culture situations in a aseptic manner in a lab called a shoot tip culture. Mostly the shoot tip culture is used to obtain the disease free plant without genetical modifications, because the shoot tips is consist of the meristematic tissues. You know pretty well the meristematic tissues is the free from the disease. It is continuously dividing cells towards the apical region or the terminal region or towards the geotropism, sorry, phototropism or geotropism. It may be differ upon the shoot or well as root. And the root tip culture, most probably the root tip culture will be used for the secondary metabolite production or some isolation of secondary metabolite or some chemical compounds, worm culture or any fungal isolation. Apical produce in vitro shoot system with indeterminate growth habits. These were among the first kind of plant tissues in 1934. Remain the importance of the research tools in the studies are developing the phenomena furtherly for another cultures like shoot culture or embryo culture and other things. An important one is protoplast culture. The first step in the protoplasm involves the regeneration of the cell wall around the protoplasm membrane. Once the cell wall has formed, the cell division must be induced the new cell. Because of the totipotency of individual cell can do new offsprings. And eighth one is the leaf primordial culture. The leaf primordial culture is the excised portion of the leaves or leaf material or leaf primordia are grown on a sterile environment. The mature leaves can be kept healthy under the culture conditions for considerable time or duration or periods. The leaf primordia have been used to study the growth and differentiation process. And you have a question, what are the stages of the micropropagation procedure or technology or any method? The micropropagation is now typically divided into 5 stages. The first stage is stage 0. The first 1 to 4 stages will be explained or proposed by Morasic. And Durbridge and Manet added the stage 0, because the stage 0 is essential to develop or to prepare the plant selection or donor selection and how to prepare the explants, because the quality of the explant and responsiveness is influenced the physiological or phytosanitary conditions of the donor plants. On stage 1 is concerned the establishment of the explant in a culture, the surface sterilization, disinfections must be free from the infections or contaminations, especially the explant tissues, from the pathogenic bacteria or microorganisms. And stage 2 is multiplication. It is the repeated enhancement of axillary shoot production encouraged by the cytokinin in a medium alone with or a small amount of auxins. The amount of cytokinin and the presence of the small amount of auxin will play a major role to determine the embryonically. The stage 3 is rooting. It is a pre-transplantation stage. If you success with the rooting, the initiation as well as multiplication of root system means that shootlet or the plantlet or the micropropagated plantlet is ready for transplantation. The advantage is the root system of the shoot cluster in vitro condition. For root initiation in vitro, IBA are important. As far as this stage 4, the acclimatization or hardening, the process by which an organism or plant physiologically as well as anatomically adjusts themselves from in vitro condition to ex vitro environment. That is the meaning of transferring of artificial environment to natural environment. So we are going to see about the some of the photographs which are related with the plant tissue culture. It may be the root culture, it may be the apical culture, leaf culture, and so many things we have collectively what we have in our lab. We trying to emphasize you can get the very clear shoot multiplication. Yeah, it is a first it is from the culture bottle, culture tube. Now we can have in it a conical flask. The hypothesis, the hypothesis, the plant, how much important, we have succeeded the in vitro propagation of the hypothesis plant with enormous shoot system. You can have the very good fleshy callus in a conical flask. You can find some of the callus or tissues may emerge the shoot system like. Wow. How the callus is regenerated or redifferentiated into a organ system like shoot initiation. Then Anicostoma, this is a very good plant, Anicostoma. We can have it. It is in a bottle, culture bottle. You have plenty of shoots. So we can calculate at least in it we have 200 numbers of shoots. The 200 numbers of shoot can be propagated with a 1 piece of or 2 centimeter piece of leaf disc. You can imagine it how the totipotency played here. And very good plant, Chlorisa superba, because we have very good photographs, we succeeded good. Then heliotropium, nice plants, have a very good stages. We can have the way, so many stages are there. Then you can have a varieties of plants. Yeah, which is the callus initiated the shoots, which is taken the in vitro to the petri dish. It is ready to subculture. Yeah, now you can see the subcultured plants. Each and every subculture plants, it can be initiated multiple. There are numerous plants. Pacoba monori, very good plant for bacchusite production. This is called acclimatization process. So, in the final stage of the plant tissue culture is acclimatization as well as transfer into the field. So, we have succeeded. Paisalis. The Paisalis plant micropropagation. There are so many stages are there. First, shoot initiation and multiplication and rooting and hardening. This is a direct somatic embryogenesis from the callus of. And finally, just we are going to see about the synthetic seed production. The synthetic seed production or artificial seed production is a very good technique to produce the synthetic seed in all plants. Instead of the seeds, we can have the synthetic seed, it can emerge or it can produce new offsprings. In that way, the method is consisting of so many steps. First, we have to select the explant from the healthy plant. We have to induce the callus. The somatic embryos will be induced in the callus. Again, it has to be transferred to the for the proliferation of the somatic embryo. After getting the proliferation of the somatic embryos, we have to allow for the maturation of the somatic embryo, and final step, we have to encapsulate the somatic embryos. By sodium alginate, and we have to do that in vitro propagation. Again, we have to acclimatization. We can induce the fruits. We can go for the again synthetic seed production. And also we should have some somatic hybridization. We are going to have it in a somatic hybridization as a separate class, but I want to emphasize the outline of the somatic embryogenesis or somatic hybridization. Somatic hybridization is nothing but the fusion of two different isolated protoplasts from two different varieties. Here the two varieties may be, one variety may be belonging to the high yielding plant, one variety may be the disease resistant plant. So in a single method, that is somatic hybridization, we are going to incorporate the two benefits or two interests of habits. And another one is So development of the hybrid plant through the fusion of two somatic embryo or somatic protoplast or two different varieties or the species, which is collectively called as somatic hybridization. This is some protocol. In the protocol, there are so many steps are there. For isolation of protoplast, we have seen already there are two methods are there. One is chemical as well as mechanical. Here I want to say one thing is, by there are in the two methods we have so many advantages as well as disadvantages. So isolation of protoplasm, one is by mechanical means. We can do some mechanical pressure on the whole intact cell, then from intact cell we have to get the intact protoplast without any disturbance. Because when you are going to apply the pressure on a cell, the breaking of the cell wall along with some disturbance will happen in the protoplast. So collection or the ratio of getting the intact protoplasm will be the minimum in the case of mechanical protoplasm isolation. Okay sir, what about the enzymatic? Even we are getting the high ratio or high percentage of protoplast from the enzymatic isolation of the protoplasm, in some places or in some plants, in some situations, the two enzymes, the pectinase as well as cellulase, may influence the protoplasm. So with the influence of these two enzymes, may the difference or variations happen within the protoplasm. Another important one is in vitro germplasm preservation or application. The germplasm, it is refers to the sum of or total of genes present in the crop or the species. It can be related to the species, it can be preserved. The conservation of the germplasm involved the preservation of the genetic diversity of the particular species or the crop or the variety. These will ensure that the availability of the valuable or vulnerable or endemic or endangered species of the germplasm to breeders to develop the new offsprings or new species or new varieties, which could be the improved variety. The germplasm may be depending upon the crop species and the method of preservation of the genetic resources from the first year, that is 1 year to 15 years. The important method of conservation is called cryopreservation. So preservation of the preservation of the genes or the gene pool of a variety or the genetic information of the variety or the species or the particular species in a cryo means liquid nitrogen. So we can preserve the number of years. It can be stored at a very low temperature, a solid carbon dioxide at minus 79 degrees Celsius, or it can be low temperature at deep freezer minus 80 degrees Celsius, or using the vapor nitrogen at the minus 150 degrees Celsius. In any cost, most recommended cryopreservation is liquid nitrogen. We can maintain at minus 180 degrees Celsius of the genetic diversity or genetic pool in any tissues or plants for the cryopreservation. For an example, meristem embryos, endosperms, ovules, seeds, and etc. Then mutation breeding, we can know that Such radiation certain the chemicals can be induced the mutation. A genetic information may be variations will be happen. It can be desired character. It should be selected. It should be preserved, or it could be breed by the breeders. Then molecular farming. Molecular farming is nothing but use of whole plants or the whole tissues or the cells cultured in vitro condition for the production of valuable recombinant proteins. Genetic engineering, you know that engineering of the hybrid or variety or any species of our desired characters which is augmented. Genetic variations in the conventional breeding. This is some production of genes. Then production of secondary metabolites. How to produce the secondary metabolites. These are some steps are there. So thank you very much. We will see in the next video.